So I've never seen the propaganda from Big Pharma or anything. I thought Russiagate was the biggest propaganda thing I'd ever seen in my life. But then when they turned on the jets against ivermectin, so ivermectin, as you know, won the Nobel Prize for Human Medicine just in 2015, but they made everyone think it was some kind of just an animal medicine. Now, 80% of antibiotics are used in cows, in farm animals. That doesn't make penicillin a fucking thing. That's penicillin's not a cow medicine. You know what I mean? So they did that. And why would they do that? Well, ivermectin is generic, so anybody can produce it. So there's no profit to be made from it anymore. And so they made everybody think it didn't work and blah, 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 even though there were 66 studies showing that and the CDC also recommended that doctors give it to people in trials to find out how it works. They recommended that. The CDC recommended that doctors give ivermectin, and they even gave a dose recommendation. And so, but you would never know that listening to CNN or listening to ABC News or anything. You would never know any of that, right? So this guy's John Campbell. He's a PhD, and I watch him for ivermectin information, and he's the one who told everybody that they're not aspirating when they do the jab, and that could, if you don't aspirate, you could shoot it into your blood vein. And there was a paper that said they did experiments on mice, and that happened. And if they put the vax into their veins, it went to their heart and gave the heart inflammation. So he was the one who taught me about that. And now he's got they have a new drug, right? They have a new drug from Pfizer that's supposed to be able to treat COVID, and he is going to put it up against ivermectin. Watch this.
A warm welcome to today's talk. It's Tuesday, the 9th of November. Now, yesterday we looked at Pfizer's new antiviral drug that shows very high levels of efficacy in preventing serious disease, hospitalization, and people dying, which was interesting. And that drug works in a particular way. It has a particular, what we call a pharmacodynamic action. It works in a particular way. But there's another generic drug called ivermectin that you might have heard of that works in exactly a same way as that. Now, no one's saying that information has been deliberately suppressed for years while millions of people have died. But what we are going to show on this video is conclusive proof from the literature that this modality of action is the same. So if you're up for that, stick around. It's not an easy watch, but I've broken it down as much as I can. Now, just before we crack into that, we need to look at what's happening. So when a virus, in this case SARS coronavirus 2, gets into a cell, what happens is it makes lots of proteins. It starts off making proteins, and these proteins are long proteins. So that's one long protein there. There's another long protein there made out of hundreds of amino acids, sometimes a few thousand amino acids, all strung together. Now, the problem is with these long proteins is they're too long for the job that's required. So it's a bit like a building site and a big log of wood arrives. It needs to be trimmed down into bits that fit in your door frames and your window frames. So this needs to be trimmed down. So these proteins need to be trimmed down. Now, how do we trim down a protein, of course, is the question. Well, it has to be done in a biochemical way. And the way it's done, particularly in SARS coronavirus 2, is there's an enzyme called 3-CL protease. Now, protease is an enzyme which breaks down protein. So this is 3-CL protease, and that will break down proteins. It's what we call proteolytic. And it will take these long proteins and it will chop them into shorter proteins. It's what we call an endopeptidase. So now instead of having one long protein, we've got two short ones, and these fit together just nicely to the new virus that we're trying to make, whereas before it was far too long, it had to be chopped. So that is the protease. Now, these new drugs are what we call protease inhibitors. They stop the protease from working. So here's my protease here. That's my 3-CL protease that chops up my proteins into the right length. And a protease inhibitor is a bit like this tape, and it's going to stop the protease from working. So there we go. We have now inhibited the action of the protease.
I don't think you needed all that tape. I think that was a lot more tape than needed. Anyway, but he definitely doesn't want it to work. So it's work. There you go.
Inhibited. So what I'm going to do now is I have a long protein here, So that one's okay. We've done that one. So that fits in nicely into the virus. But now there's another long protein here that needs processed. So the protease, the 3CL protease, comes along, ready to chop this up into shape. And, I can't open it now. So what we've done is we've bounded up the active site of the protease. And that's what these drugs do. They bind the active site of the protease and they stop the protease from working. That means they stop the protease from chopping up the big proteins into smaller proteins, into these strings of amino acids, so they can't build the virus. So it inhibits viral replication, these protease inhibitors. So that's kind of what is happening here. Now, this is the new Pfizer.
So the protease inhibitors stop viral replication. Okay. All right. I mean, if you do that, if you inhibit the protease, you would stop viral. You already said.
Here that's just come out. New Pfizer antiviral. We're going to compare it with ivermectin in this video. A pharmacodynamic analysis by me. Pharmacodynamics is how the pharmacy works, how the drug works in affecting the body. Now, this is the new Pfizer molecule here. This is the new drug. And this is the shape of it here. So it's got 3 fluorides there. So it's interesting. That's the shape of the molecule. Fair enough. Now, this is a new molecule. Well, certainly on me, but I'm no molecular expert. But so here we have, for example, ritonavir, which was an old antiviral drug that had been using since 1996 and which in fact will be given with the new one to work together. So we can see that they're different shaped molecules, can't we? There's no F's on this one, for example. So that's the different molecule. So if this molecule was patented and this is a new molecule, this molecule will be out of patent, even though it has similar actions.
He says patent. He means patent. He doesn't know how to speak correctly. He has too much vitamin D in his head.
Then if we look at ivermectin, which is also out of patent, of course, can't make any money out of ivermectin.
Can't make any money on it.
Then where is it? There we go. So that's the ivermectin molecule there. And we see that's a completely different shape to this new Pfizer molecule here. So they're completely different looking molecules. So as a completely different looking molecule, I would imagine that that's absolutely fine to patent that because it's a new molecule. And when you pay...
So that's the Pfizer new drug to treat COVID, and he's showing how it's different molecularly than ivermectin. So he said it should be okay to patent that. So now they have a patent on this new drug. Okay.
Drug, you can make money out of it for the next 20 years.
A lot of money you can make out of that.
In most jurisdictions. Of course, by the time the drug gets to market, it's often a bit less than that, but that's the sort of time when you can make some serious money out of these things. Now, first of all, this is the new Pfizer drug here, the PF0732, is designed to block the activity of the SARS coronavirus 2 3CL protease. 3CL protease. So that 3CL protease now won't work, it won't open, so I can't chop my proteins into the correct length to build a nice new virus. Now, evidence for that, because we always have to give the evidence for that is in this paper that we looked at yesterday. So check it out, and there's evidence for that there. That is them saying exactly how this...
So, well, let me just back this up a second. Hang on.
The evidence for that is in this paper that we looked.
So look what this paper says. Pfizer's novel COVID-19 oral antiviral. This is the drug we're looking at now as compared to ivermectin. This has reduced the risk of hospitalization or death by 89%. So they have a drug that, according to them, works 9 out of 10, 90%. Wow, that's unbelievable. And because if you keep in mind, the rate of hospitalization from COVID is under 1%. So this reduces that by 89%. So now what is your real risk of hospitalization if you have this drug? Okay, so what I'm saying is there's therapeutics out there to treat it. Okay, here we go.
Yesterday, so check it out, and there's evidence for that there. That is them saying exactly how this new drug is going to work. That's the site there. So we now know what a protease is. It breaks down proteins into the right length. A protease inhibitor stops the protease from doing that, therefore the proteins remain too long. And a 3CL protease is a protease that makes SARS coronavirus 2. And of course, a 3-CL protease inhibitor will stop it from making SARS coronavirus 2, therefore is antiviral. Now, I'm pretty sure that all made sense. So if you haven't quite got that bit, do just go back and watch it again, because that's how it works. Now, the CL, this stands for chymotrypsin-like protease, 3CL, 3-chymotrypsin-like protease. Now, everyone, of course, in human biology has heard of chymotrypsin. It's a pancreas release. It's an enzyme released by the pancreas to digest protein. It chops proteins up. So it's a protein chopping up enzyme. So this this protein, this chymotrypsin-like protease inside the virus, is working in a very similar way to the chymotrypsin that your pancreas produces to digest your proteins. Now, going on to the first evidence I want to give. Now, the first evidence comes from this paper here. So we're going to be looking at this paper here, available. You can check it out for yourself. I always paste the links, of course, because you don't want to believe what I say. We have to go by the evidence. Identification of SARS coronavirus 2, 3 chymotrypsin-like protease inhibitors, which we now know. Remember, the inhibitor is the piece of sellotape that stops the enzyme from working, or the scotch tape, or whatever you call it. That's the sticky tape that stops it from working. That's what's clogged this up, and I just can't work these scissors now. Completely useless to chop up my proteins. So this is a quantitative high throughput screening. So this was looking for compounds that could do this. This was done back on the 3rd of September 2020. So here they screened all these compounds. They did a high confirmation screen. They did a finer screen for these compounds, and the activity of anti-SARS coronavirus 2 viral infection was confirmed in 7 of the 23 compounds. So they found 7 compounds that were worth looking at that would have this protease inhibiting effect. And this paper also helpfully gives them.
So just so everybody's, so you get it. So to make the SARS-CoV virus, you have to break a protein. You got to cut it. So this drug stops the thing that breaks the protein. So you can't replicate the virus. So now they're looking at drugs that do that. And 7 of the 23 compounds did that. And now they're going to look at it closer. Here we go. Ready?
Explanation of it, hopefully the same as mine. But there it is here. So here's the chain of amino acids forming the protein. But that one's far too long, so it's got to be chopped by that little pair of scissors there, which is the protease. That's the scissors of the protease, of course, and it chops it up into 2 bits, which are nice virus-sized protein chunks that can be used in the door frames and window frames or whatever anatomical molecular architecture is required for the virus. So that was screening compounds. Now, next there was a lot of papers on analysing these. So this is microscopic interactions between ivermectin and key human and viral proteins involved in SARS coronavirus 2 infection. Now, obviously, you want the evidence for this. You're not going to take my word for it. Here it is. It's on this paper here from no less than the Royal Society of Chemistry. And again, thankfully, all these are available. And if you're a biochemist, you'll understand that. If not, you'll just get the gist the same as me. But I think we have got the gist. Now, that's the reference for that one there. The strength and persistence of the interaction between ivermectin and the binding site of the 3CL protease indicate that a partial inhibition of the catalytic activity, in other words, the way that the enzyme works, could have a place as the drug interactions with the main subdomain that define the enzyme define the enzyme binding pocket. Now, the enzyme binding pocket, of course, the protease is an enzyme. So what it's saying here is, so the enzyme's obviously not like a pair of scissors, but if we imagine it's like a pair of scissors, so imagine it's got kind of a blade there, and it's got a bit goes around there, and kind of another blade there. Well, the enzyme binding pocket is that the substrate will bind into that bit there and hold the substrate while it gets chopped up. But if you block that up with something, so for example, if you block that up with ivermectin, that no longer works. That's what it means. So it's telling us how this is working to block the activity of the 3CL protease. That's what that paper's about, and it makes perfect sense if you do take a bit of time to read it. Now, the next paper is this one, again giving you all the evidence, identification of 3 chymotrypsin-like protease inhibitors as potential anti-SARS coronavirus 2 agents. Don't worry if you're not getting all the terminology. I don't always get it myself, but that's this paper here. Out of 13 OTDs. Now, OTDs, this is called off-target drugs. So ivermectin wasn't originally one of the antivirals they were looking at. They were looking at other existing antivirals. So that's why they're calling it an off-target drug there. But out of 13 off-target drugs, only ivermectin completely blocked. Actually blocked by 80%. The three...
So again, the people who are saying that it's crazy, it's an antivirus, it doesn't work for this, it doesn't work for that's for parasites. What do you do? So there it is. Of the 13 of those off target drugs, only ivermectin completely blocked 80% of those protease, and there you go. Whoa.
CL protease activity.
And it's inexpensive
at this particular concentration of ivermectin. Now, if we look at this compared to the other drugs, this is particularly useful. So here's all the other drugs they were testing along the bottom here. So they were testing all of these medicines along the bottom here. That's what they were testing. And this is the percentage of 3-CL enzymic activity present. So the shorter these bits, the more the enzyme was inhibited. So we see that this here, GC3227, that inhibited the enzyme a lot. So the enzyme couldn't chop up the proteins. I don't know what that is. It's not mentioned because it might be toxic. Anyway, all these other ones didn't inhibit it very much, but ivermectin inhibited it a lot. So the ivermectin from this study is greatly inhibiting the ability of the pair of scissors, the 3CL protein, to open. Therefore, it can't chop up the proteins. Therefore, you can't make new virus. So that is what that paper is saying.
So that paper is saying ivermectin works to fight COVID. That's what that paper is saying. It inhibits the replication of the virus.
Validation and approval of COVID-19 specific drugs takes years, this paper also says. Therefore, the idea of drug repurposing, drugs that are used for other purposes, also known as repurposing, is an important strategy to control the sudden outbreak of life-threatening infectious agents that spread rapidly. We agree completely. Drugs.
So what they're saying is that, well, development and approval of a drug will take years, but you could take a drug that we already use and we know isn't toxic, and it's already approved, and you could repurpose it. There it is. That's an important strategy. So doing something like they're doing with ivermectin is an important strategy. That's what this says. And it has against life-threatening infections and agents that spread rapidly.
Be repurposed much more quickly than we can develop new drugs, because we already have a pretty good view of the safety profile, the pharmacokinetics and the pharmacodynamics of the drug. The kinetics being the way that the body interacts with the drug, and the dynamics being the way that the drug affects the body. Right, next paper. Next line of evidence here is from this paper here. Again, Royal Society of Chemistry, no less. And all the details are there on that paper as well. So again, feel free to check that out if you would like to. It's all there. Let me give you the gist of what it's saying. From the docking analysis, that's the way that the drug docks into the enzymes, the way that the drug binds up the scissors in this case. From the docking analysis, ivermectin showed the highest docking score with an average energy of minus 8.5 kilocalories per mole. I don't know the units. I don't pretend to know it. But it means it bound on quite tightly among all compounds. So it showed the highest docking score. In other words, it cluttered up the enzyme. It cluttered up the 3CL enzymes and stopped it working better than all of the other compounds that they looked at. Remdesivir showed the lowest binding energy and highest docking score. So remdesivir doesn't work very well now on this particular analysis anyway.
Which is what Dr. Yoganda said when he was on this show. He said that the remdesivir is a crime. And he wanted to talk about how that is, of course, I'm sure you can make $1 million off it, which is why they're pushing it.
Now, here we have some information about remdesivir, which, of course, has been used for critically ill patients quite a lot. I put this up here because we notice it's £340 a vial. £340 a vial. That's the indicative price that the UK is paying.
That's the price, which is roughly around, what, $600-700 US, right?
So that's the link for that from the National Institute for Health and Care Excellence. So pretty expensive drug. I believe in the United States, a course costs about $3,000 for a 4 or 5 day course? Can't remember. Anyway, really expensive. And of course, we know ivermectin is available through the World Health Organization at 6 cents. 6 cents.
$3,000 versus
5 English pennies a tablet.
Huh.
So it's a bit cheaper.
Bit cheaper.
And from this data, higher docking score, higher
and a
cluttering up ability.
And it works better.
Of the enzyme. Moving on to the next piece of evidence. We've only got a couple more, so stick with it. It is worth it. This is from Future Virology. So this evidence comes from exploring the binding efficacy of ivermectin against key proteins of SARS coronavirus 2, pathogenesis, and in silico approach. Now, this is very interesting. In silico means this is computer modeled. Now, this is incredibly clever and way beyond my ability. It's computerized molecular modeling, but it is a very well recognized science these days. So let's see what this is saying from the future of virology. We have documented an intense binding of both ivermectin B1A. That's the type of ivermectin in the drug, and that's the type of ivermectin that's similar, but not in the tablets. But these are the two natural forms that were first identified by William Campbell and Satoshi Amuro, for which they won the Nobel Prize. So we've documented an intense binding effect of both ivermectin and this to the main protease with a high binding energy is all that means. In other words, the ivermectin well and truly clogs up the 3CL protease, stops it from cutting up the long protein chains into smaller protein chains. Therefore, it stops the viruses being made. So just as a reminder, given that with this evidence, this was from the original Pfizer paper, it is working this way because the original Pfizer paper says that the new Pfizer medication is designed to block the activity of the SARS coronavirus 2, 3-chymotrypsin-like protease. In other words, it acts the same way as the sellotape round the pair of scissors, as indeed we have given evidence for ivermectin doing. Now, we could give many other examples. I might just give... I'll give one other brief example here, I think, because I don't want to get too cluttered. So just briefly, this evidence here is from this paper here. Always give the evidence. Frontiers in Microbiology, published in January 2021. Again, all the information there, and you can download the PDF, download the article. It's great that these are all freely available, which is just brilliant. Molecular docking reveals ivermectin and remdesivir as potential repurposed drugs against SARS coronavirus 2. This paper said, don't take my word for it, check it out. That's what it says. Right, with SARS coronavirus 2. Now, first of all, the ivermectin inhibits the spike protein. This is saying. So, as we know, the spike protein in the virus here, the spike protein has to fit into the ACE2 receptor site on the cell surface in order to get into the cell. So this is saying that the ivermectin clogs up the spike protein. It no longer fits because that's got to be a perfect fit. So with SARS coronavirus 2 spike protein, ivermectin showed a high binding affinity. In other words, that hung on good and tight. It bound it, and that would stop the spike protein from infecting the cell. Ivermectin showed a high binding affinity to the spike protein, as well as the human cell surfaces receptor of the ACE2. So, and that's the other one that it can bind into there. So, in other words, not only does it clog up that, it clogs up this as well. Therefore, it's a double reason not to fit. So it doesn't work just one way, ivermectin. It works two different ways, is what he's saying. It screws up the spike protein and this thing receptor here. Double screws it up. So you can't get the COVID. In other words, this is the key here, isn't it? That's the key. And this is the lock. So not only does ivermectin bend the key, It also stuffs plasticine into the lock, would be another way to do it. So you're not going to get the binding there. So that's interesting molecular intimation of possible pharmacodynamic modalities of efficacy. In agreement with our findings, ivermectin was found to be docked between the viral spike and the ACE2 receptor. Well, there you go. In other words, it clogs that space up there. So you don't get the binding. Therefore, the virus can't stick its RNA into the cell. The RNA cannot penetrate. The viral RNA cannot penetrate the cell because it has to dock first in this process called adsorption. In agreement to our findings, ivermectin, I've said that, so it clogs up both. Now, it also binding interactions of selected drugs with human ACE2 protein, other ACE2 proteins as well. So it's binding up other ACE2 binding proteins. I'm not going to go into the detail; they're in the paper if you want it. Ivermectin also binds with interaction with selected human ACE2 proteins, as we've said, but that's another sort of set of proteins. With SARS-2 glycoproteins, ivermectin has the highest binding efficiency to the predicted active site of the protein. In other words, good, so it's clogging up this protein as well, which is great. And again, this other protein here, NSP14, don't know what it is, but ivermectin clogs it up. Shows the highest binding affinity.
Wow, it's a lot of stuff that ivermectin
binding
clogs
interaction
up.
with selected drugs with SARS coronavirus 2 PL protein. I don't know what that is either. But again, it clogs it up. So whereas the Pfizer drug is only working, as far as we've been told in the Pfizer press release, against one biochemical modality of viral replication, the ivermectin is working at many different levels. Now, the fact that the Pfizer medicine is only working against one particular biochemical pathway means to me that the virus could learn to avoid that. It could evolve to be drug resistant, as indeed the early antiretrovirals did with HIV. So that's possible with ivermectin because it's working on so many different levels. That the idea that a virus would mutate in a dozen different ways to avoid all those different mechanisms, or whatever it is, six mechanisms we've talked about today, I think. The idea that we get 6 mutations that could dodge all of those all at the same time is improbable, to put it mildly.
So he's saying the new Pfizer drug only inhibits the creation of the SARS-CoV in one way. Turns out ivermectin does it in many different ways and many different levels. So the chance of the virus being able to get around ivermectin and mutate to get around it is very low. But to get around the new Pfizer one, he's saying it looks like it's probably pretty high that it could do that. Just like with the antiviral drugs they originally had for HIV. They got around it real fast. Well, this ivermectin blocks it in 6 different ways. So the idea that it could get around it probably won't. Isn't this fascinating? Why don't you know this? Because ivermectin's cheap. It's 6 cents a pill, like you said. And nobody can make money off it. And you live in a world that is nothing but 100% corporate propaganda.
So I've got a message here for world leaders, a brief message to world leaders, people that are making the decisions about this. Come on, you all. You're not a horse. You're not a cow. In other words, world leaders, you're not a horse, you're not a cow. Come on, you all. You've got a human intellect. Let's use it to follow the scientific evidence to save human pain, suffering, and death. Thank you for watching.
So what he's doing is mocking the FDA's tweet when they said, hey, y'all, you're not a horse, you're not a cow, don't use ivermectin. He's mocking that. He's telling you, hey, world leaders, you're not a horse, you're not a cow. Use your intellect to follow the science and not the propaganda around ivermectin. Isn't this amazing? Isn't this stunning? It should be, because you've been propagandized. You've been propagandized. There's a cheap and effective treatment out there according to these papers. And it stops the replication of the COVID virus, according to these papers. This country is the most propagandized country in the world. Our government is completely owned by corporations, and so is our media. That one paper he talked about was from January 2020. So this is a secret. People know this. The people who are pushing All the propaganda around ivermectin know this. The people at Big Pharma, they know this. Sanjay Gupta, he knows this. Now you know why people get their news from YouTube. I get my news about ivermectin from this guy on YouTube. I'm certainly not going to go to CNN. I'm not going to go to MSNBC or the Washington Post or the New York Times, which is funded by Big Pharma. I'm not going to them. I'm going to go to this guy. He's a PhD, and he knows what he's talking about, and he doesn't have an agenda. And by the way, he shows his work. So if you got a problem with him, then you got to go argue with those peer-reviewed studies. Yeah, I know. Hey, come see us do a live stand-up show in Portland, Oregon, the 26th and 27th of November. That's Thanksgiving weekend. See you there. And please join our premium program. Get a lot of extra stuff. We'll talk to you later.